Hi, I'm Dr. Divyashree Gangaraju, heading the Bioprocess Research Division at IOM Bioworks Private Limited, Bangalore. In previous lecture videos, we have seen the definition for probiotics and its history. Taking this subject forward, in today's class, we would be discussing about the sources from which we can isolate probiotics and what are the methods for its characterization. The sources from which we can characterize probiotic microorganisms can be broadly classified into two categories, of which the first one is known as conventional sources, from which we can isolate probiotics. The conventional sources are the sources which can be relied on for the isolation of probiotic microorganisms, which includes fermented vegetables like kimchi, fermented pickles, dairy products like yogurt, kefir, cheese, and buttermilk, which are rich in probiotic microorganisms as they have been used during the fermentation process. We can also use or consider human and animal gut samples such as human feces or the animal feces for the isolation of the potent probiotic microorganisms, which shows better adaptability towards human GIT or animal GIT. Breast milk also contains significant proportion of probiotic microorganisms, and this can also be viewed as a sample or a source for isolation of a potent probiotic microorganisms. The second category is the non-conventional sources from which we can isolate the potent probiotic microorganisms, which includes the plant-based fermented foods, soil and plant root environments, marine sources such as seaweed and fish intestine, and we can also rely on honey and honey-based products in order to isolate the novel probiotic microorganisms. Under non-conventional sources, we have a first category of products which are known as plant-based fermented foods from which we can isolate these probiotic microorganisms. The examples of plant-based fermented foods includes miso, temp and natto. So these are the fermented food products which are prepared using plants as a base material. Miso is a Japanese traditional fermented food prepared using soya bean, which is fermented using Aspergillus oryzae. The temp is again an Indonesian based fermented product prepared using soaked and cooked soya bean, but instead of fermenting with Aspergillus oryzae as in case of miso, this temp is being fermented using Rhizopus, the mold Rhizopus. Natto is also a classical example for plant-based fermented food from which we can isolate probiotic microorganisms. This natto is again a Japan traditional food product which is prepared using soya bean but fermented using an organism called Bacillus subtilis subspecies natto, and hence it got the name natto. So these are the three classical examples of plant-based fermented food products which serve as a source for the isolation of unique probiotic microorganisms. The next category of non-conventional sources is the soil and plant root environments from which we can isolate the microorganisms such as bacillus, which are also known to offer gut-specific health effects. We can also rely on marine sources such as seaweed, macroalgae, which contains the probiotic like microorganisms which are known to offer specific health benefits. We can also rely on fish intestine from which we can isolate unique probiotic microorganisms which shows better adaptability for the aquatic microorganisms. At last, we can also use honey and honey-based products which might contain few beneficial probiotic microorganisms. Therefore, the isolation source for the probiotic microorganisms can be viewed from conventional sources as well as non-conventional sources. Now, once we have these conventional and non-conventional sources, the next step would be isolating the procedure. Once we have identified the source from which we can obtain the probiotic microorganisms, the next step would be isolation procedure. Now, to isolate the probiotic microorganisms either from a given conventional source material or a non-conventional source material, the first step would be sample collection and preparation. Now, to collect your sample, which could be either conventional source or a non-conventional source, you should perform the collection procedure in a sterile conditions in order to avoid contamination. Now, once you have collected these samples under sterile technique, the next step would be serial dilution. In case if you're dealing with a liquid sample, say for example, you have got honey-based products, which is liquid in nature, or it could be a marine water, which is again liquid in nature, which can be viewed as a source for isolation of probiotic microorganisms. In that case, if your sample is liquid, you can directly go for serial dilution procedure. But in majority of the cases, you might be dealing with the solid samples. It could be fermented solid food material. In that case, what you have to do is you have to adopt another technique known as homogenization. The purpose of homogenization is to get the homogeneity of a solid sample which you are dealing, which aids in the release of probiotic microorganisms into the medium from the food material. And once we perform this homogenization procedure under again a sterile conditions in order to avoid the contamination, and while performing homogenization procedure, you might require a sterile water, the water which is sterile, not being contaminated with any microorganisms or the bacteria, or we can also use PBS, what we call it as phosphate buffer saline, in order to aid the dilution of the solid food material, which helps us with the homogenization procedure. Once we have collected the solid food material, which can be viewed as a source of probiotic microorganisms, that will undergo homogenization procedure in order to get the homogeneity or the homogeneous liquid suspension, which is further diluted using either a saline or a phosphate buffer saline or a sterile water. And then the next procedure would be serial dilution. I hope all of you are aware of the principle and the procedure behind serial dilution, wherein you take 1 gram of sample or 1 ml of the sample and then dilute with 9 ml of the sterile saline, which is then serially diluted in order to achieve the required dilution factor. Now, from the each diluted plate, so each diluted tube, you would be taking 100 microliter of the sample in order to serially plate onto a medium on which you can grow or isolate the probiotic microorganism. Now, once we have serially diluted our sample for the isolation of the probiotic microorganisms, the next step would be plating these serially diluted samples onto an appropriate medium so that we can grow and isolate the potent probiotic microorganisms. The medium, which is generally used for the isolation and the growth of probiotic microorganisms, is known as MRS medium. MRS stands for D-man rugosa Shab, which was invented or developed in the year 1960 by the 3 microbiologists named D-man, rugosa and Shabt. That is why the medium is named after their initials called MRS. If you see the composition of this MRS medium, which is as displayed in the slide in a tabular column, it comprises of both macronutrients and micronutrients. I'm sure all of you know the difference between macronutrients and micronutrients. Macronutrients are the nutrients which are used while formulating a bacterial medium at a concentration above 500 millimolar units. Micronutrients are the nutrients which are used while formulating the medium at a concentration less than 500 millimolar units. So as the name says, macronutrients are the nutrients which are required at a much higher quantity, and micronutrients are the nutrients which are required at a much lower quantity. If you see the composition table, you have carbon source as dextrose or glucose at a percentage of 2%, which is equal to 20 grams per liter. You have other nitrogen sources which are used at the macronutrient level, which includes protease peptone, beef extract at 1% level, yeast extract at 0.5%, polysorbate 80 or tween 80, which is used as a surfactant, ammonium citrate, sodium acetate, again as a macronutrient level. We also have micronutrients, which includes magnesium sulfate, manganese sulfate, ferric ammonium citrate, which are used at much lesser quantity in comparison to the macronutrients. And if it is MRS agar, we would be using agar at a proportion or the concentration varying between 15 to 20 gram per liter or 1.5 to 2%. Esculin is used at 0.3%, which aids in the selection of an appropriate probiotic or the lactobacillus strain from an isolation source. So this MRS medium can be viewed as a selective culture medium in order to isolate and grow lactobacillus species and other lactic acid bacteria. Now let us assume we have got the source from which we have to isolate the probiotic microorganisms. It could be conventional sources or non-conventional sources. And again, it can be liquid in nature or a solid in nature. If it is liquid in nature, we can directly perform serial dilution procedure for the isolation. If it is solid in nature, we need to adapt homogenization technique in order to have homogeneity in the sample, and post that we would be performing serial dilution procedure. Once we perform serial dilution procedure for liquid as well as the solid samples, we would be cultivating or spreading these samples onto an MRS plate, which is a selective medium which is used for the cultivation of lactobacillus and other lactic acid bacteria. Let us assume we have performed all these steps sequentially and we have incubated these spreaded plates at a temperature of 37 degrees Celsius in an anaerobic incubator for about 24 to 48 hours. Post this, you are able to see a colony on a plate. So now what does this colony indicates? It indicates that the grown colony might be a lactic acid bacteria or a lactobacillus in nature. Now these colonies, we need to be picked up individually, or we have to select these colonies and streak it onto another plate in order to have multiple solid plates, or it can be taken onto a broth medium. Again, what is the broth medium? It's again an MRS medium which is devoid of agar. If it is MRS agar, we would be using agar at a proportion of 1.5 to 2%. If it is just MRS broth, which is a liquid medium, this is again contains macro and micronutrients as we discussed in the previous slide, but this MRS broth is the devoid of agar. Now once we have picked the individual colony from a plate and then inoculated onto an MRS broth, we would be incubating this broth again at 37 degrees Celsius in an incubator for about 24 to 48 hours. Now, once we have got the liquid sample from a solid culture, what we would be from a solid plate, the next step would be characterization of the isolated culture. We should make sure that the culture which appeared on an MRS plate belongs to a lactic acid bacterial group, right? For which we need to perform few characterization procedures in order to make ourselves sure that the culture we are using or dealing is a category of lactic acid bacteria. The first step, which is used for characterization of any culture, not only probiotic culture, is known as gram staining. I'm sure all of you know the principle and the procedure behind gram staining procedure. These lactic acid bacteria or the probiotic bacterial group belongs to a category known as gram positive. So if they are gram positive, they have to appear violet in color on microscope once you perform this gram staining procedure. And this gram staining procedure can be viewed as a preliminary identification procedure. The next step, which is used to confirm or tentatively confirm the probiotic microorganisms, is the catalase test. The first step would be gram staining. You should get gram positive result because these microorganisms are gram positive in nature. The second test is known as catalase test. What are catalase tests? The catalase tests are the test which confirms the production of an enzyme called catalase by the microorganisms which you are dealing with. These catalase enzymes, what they do, they hydrolyze the substrate which is known as hydrogen peroxide, H2O2, and decomposes into H2O and oxygen. Now, if the probiotic microorganisms produces this enzyme catalase, it converts hydrogen peroxide into water and oxygen. As the oxygen is being liberated, there would be effervescence in the sample, as you can see in this slide. Effervescence meaning the bubble formation. Now, these probiotic bacteria or the lactic acid bacteria are gram-positive but catalase negative, meaning they cannot decompose hydrogen peroxide into water and oxygen. So, if you're tentatively dealing with lactic acid bacteria, ideally you shouldn't be seeing effervescence formation, or there shouldn't be any bubble formation. Once we have done preliminary identification using gram staining test and catalase test, we don't stop there, right? Those are the preliminary identification procedures which can tell us, okay, the culture you are dealing with might be a lactic acid bacteria or might belong to a group of lactic acid bacteria. We need to be ourselves so sure in order to have better applicability. For that, we should be performing genome identification procedure, for which we would be relying on 16S rRNA amplification procedure. So we all know the prokaryotic ribosomes are made up of two units, which includes 50S and 30S, comprising of 70S ribosomal units, or they make up 70S ribosomal units. This 50S ribosomal units comprises of two components, which includes 5S rRNA and 23S rRNA ribosomal units. However, 30S ribosomal units, which is a small subunit of the 70S ribosomal unit, it is just made up of one component and what we call it as 16S rRNA component. I hope I'm clear here. The prokaryotic ribosome is made up of, or is called as 70S ribosome unit, which comprises of 50S and 30S. 30S contains just one subunit of 16S rRNA. However, 50S ribosomal unit, which is the large subunit, comprises of two components, which includes 5S rRNA and 23S rRNA ribosomal units. But our focus is on 30S, which is the small ribosomal unit, which comprises of 16S rRNA subunit. If you see the structure of 16S rRNA subunit, it comprises of two regions. The one is conserved region, the other one is variable region. What are the conserved regions? These are the regions which are conserved through the evolution. And if we design primers to amplify these conserved regions, it acts as a marker or a path through which we can identify the bacterial species. Now, keeping this logic in mind, we would be designing primers to amplify a part of 16S rRNA in order to aid in bacterial identification procedure. Now, to perform this genome amplification procedure or 16S rRNA amplification procedure, the first thing what we have to do is we have to take those tentatively called probiotic microorganisms. You remember, we got it from food sample and now we have it in the broth medium, right? What broth it is? It is called as an MRS broth. So now from the broth you centrifuge in order to suspend these bacterial cells as a pellet, wash the pellet and then perform DNA isolation procedure. Using phenol chloroform method, you can isolate the DNA from the given bacterial cells, or if you have a DNA isolation kit, you can isolate the DNA from the DNA isolation kit. Once you have the DNA isolated from the bacterial samples, use specific primers in order to amplify the 16S rRNA gene from the bacterial sample. And once you perform this amplification procedure using polymerase chain reaction, the next step would be running the gel electrophoresis technique in order to see if you have got the band at the expected size. If you have got the DNA band at the expected size, it indicates the success in the amplification procedure or the PCR procedure. And once you are sure of the amplification procedure, you have to excise the band from the gel electrophoresis unit and then purify the DNA and then give it for sequencing. And then you have to analyze the sequencing data, what you have got in ADGC format, using a suitable bioinformatics tool. This is the blast results display you might get once you blast the query of the sequence you have got through sequencing machine. As you see in the table, you have 100% similarity of your query sequence with a lactobacillus plantarum. However, you have very less percentage similarity with other bacterial genera or the species. So this indicates that the isolated microorganisms belongs to a genera Lactobacillus with species Plantarum, as it is showing 100% similarity index with just Lactobacillus plantarum. So this is how you name the probiotic microorganisms. So far we have seen the different sources such as conventional and non-conventional sources from which we can isolate probiotic microorganisms using a specific isolation procedure, and the preliminary identification procedures such as gram staining and catalase, as well as 16S rRNA genotypic identification procedure in order to characterize the probiotic microorganisms. Let us stop here, and in next class let us discuss about the different aspects of probiotics and its classification. Thank you.